I'm so sorry, Haley. I forgot to start the Zoom. We're here now. It's not your fault. Just email me, Haley, and I will get you the quiz that you missed. That was my fault. Okay. All right, so starting back up with the lecture. We have our MHC molecules telling you that I found some kind of antigen from either outside or inside of the cell, right? Then your B cells or your T cells have special receptors on them. The receptors on your B cells and T cells, they don't just recognize any antigen. Each B cell and each T cell, those receptors that are on there that we can see here, this or this on the T cells. This is B and this is T. Those receptors that are there, they only recognize a specific antigen. They don't recognize any antigen. So I could be a macrophage that picked up something from outside, had it from outside. I put it on my MHC2, right? This came from outside. And I'm saying, hey, on my handle, my MHC2, I found something. And that thing might encounter a million different T or B cells. But until it finds the one that recognizes this marker, right? That specific antigen, so black top marker, until it finds that specific antigen, it doesn't engage. All right. Then we have these other things that are going to help make sure that we are going to be able to elicit the proper response that are hanging out at the sides of these guys as well. And we're going to learn a little bit about those too. But those receptors are what's going to be interacting with that specific antigen. So once that B or T cell comes into contact with something presenting antigen or with antigen itself, that receptor on the B cell or the T cell only recognizes a specific antigen. So you have 8.6 billion different B and T cells that can recognize 8.6 billion different antigens, and we're trying to find the right one. Some of those got eliminated because they reacted to self early on in their development. So we already narrowed it down to less than 8.6 billion. But there's still so many others. Even if it's just 1 billion left, 1 billion cells in your body, just looking around, and that one has to find the antigen. So it could be floating freely, and the B cell can interact with it that way, or it could be presented on MHC. All right. So somehow these receptors encounter their antigen. They have sites at the very tips of their receptors called antigen binding sites. So those are going to be extremely variable. And the way that we can get the ability to recognize 8.6 billion different antigens from just your 20,000 genes in your entire body actually comes from these specific regions called VDJ segments in your genes. And we can just reassort different V's and D's and J's and then insert random, literally just random nucleotides in the reassortment process to create all this variety that we have as a result. Each receptor, especially on our B cell receptor, so that's basically our antibody, right? It's got a variable region and a constant region. The variable region is the one where we get the variety. That's where we're going to be interacting with our antigen. The constant region, it doesn't have any variety in it. That's why it's called constant, right? You can rely on it to always be what it is. And that constant region is what dictates the function of the antibody or immune globulin. Okay, so all that's dictated by the constant region. It looks like a handle at the bottom of the things. And at the top in our variable region is where we're interacting with all those, like one antigen for each antibody that we make. Okay, and that's where that very specific interaction is going to happen. These are going to consist of a light chain and a heavy chain. So what this is actually talking about is when you draw an antibody, this is the heavy chain. All right. And then you have these little pieces out to the side, those pieces out there. These are the light chain. And that's basically the structure of the antibody there. The T cell receptor is very similar. It's not shaped like the Y the same way, but it still works very similarly. It's got a more variable region at the end that interacts with antigen. And then we have a portion that's attached to the membrane. Our immune globulins in our B cells, we have versions that are stuck in the membrane as a receptor, and then we have versions that we secrete that are just protein fluid, and it's our antibody, right? So the antibody and the membrane-bound portion. T cells only have membrane-bound portions. So T cells only have their T cell receptors on their membranes, and that's all they're ever going to be, is on their membranes. So in T cells, the T cell response, because we're not making something that goes out and fights for us like the B cells do. The B cells, the bombers, they make bombs, which are our antibodies that are going out and doing the work. The B cells aren't doing anything else otherwise. They're just making antibody, and the antibody is serving the function. T cells are actually doing the work. So the T cells are either going around interacting with cells, telling them, yes, you may go ahead and respond, or they're going out and killing infected cells. But the cells themselves, the T cells themselves, are doing the work. So we call the T cell response the cell-mediated response because cells are doing the work there. We call the B cell response the humoral response because in your humors, in the fluid in your body, the antibodies are doing the work. Okay, so that's the distinction there with those guys. All righty, so this is going to talk a little bit about those varieties. You might have all of these different genes that are associated with ability to interact with antigens in general. And then we can mix and match the segments to get the variety that we get in each antibody. A little video on this. I do want to play it really quick because I just think it's super cool.
People are full of antibodies, a lot of antibodies. These proteins are a vital part of the immune system, but just how the body makes such a variety of them mystified scientists for decades. The human immune system has to fight off an endless variety of bacteria, viruses, and parasites, and for each invader, it needs a specific antibody. Luckily, it can generate billions of them, each slightly different, ready for any disease. But these antibodies, made from 2 light chains and 2 heavy chains, are proteins, and billions of different proteins should need billions of genes to make them. Humans have around 20,000 genes. The solution to this problem was a Nobel Prize-winning discovery. Antibodies are made by B cells. Like every other cell, B cells start off with the same 20,000 genes, but they also have an enzyme called RAG. RAG is a DNA shuttler. It targets the genes that make the antibody's binding site. There are 3 kinds of gene segments here, called V, D, and J. For the heavy chain of the antibody, humans usually have around 130 different V segments, 27 D segments, and 6 J segments. The RAG enzyme binds to one of the J segments at random, and then to one of the D segments, chopping off the spare DNA between them. Other enzymes then join the ends together. The same thing happens with a random V segment. So you end up with a gene composed of 1 V, 1 D, and 1 J joined together. This provides over 20,000 possible V-J combinations for the heavy chain. The light chain can have another 400 possible gene combinations. Combining heavy and light chains gives approximately 8 million different possible antibodies, each with a different binding site. And that's not all. During the joining, extra DNA is added or removed at the junctions of the segments, which takes our antibody diversity from the millions into the billions. This is how our immune system can respond to diseases it's never even seen before. In fact, there's so much diversity, one of our antibodies could potentially bind to almost any target in the whole universe.
So pretty cool stuff. I like the visuals of it. Alrighty, now I gotta get out of this. All right, back into this. All right. Alrighty, so if we can have all of these cells that can make all of these different antibodies or react to all these different antigens, then we need to be able to basically choose which one's going to proliferate at any given time, as well as eliminate any of those that recognize self. And that is what clonal selection is going to get into. Basically, a clone, we kind of already know what clone is, making a copy of the original. Well, we're going to proliferate a particular lymphocyte, clone that lymphocyte out whenever we choose it to. And then those clones will differentiate into the different working types involved. So we have all this clonal diversity at first, we'll need to eliminate any of the self-reactive ones, and this happens very early on in our development, and this is called clonal deletion, removing the anti-self guys. Remove anti-self. We'll get those getting rid of very early on in our development. And so this leads to immune tolerance. This means that we tolerate our own antigens. That's really what that's referring to. So now we have a whole repertoire of lymphocytes left that shouldn't be anti-self, that could potentially interact with any antigen ever, as long as it's non-self. And we have one cell essentially for each antigen possible. And once that one cell interacts with the antigen, and specifically the part of the antigen that it interacts with called the epitope, once we've engaged with that antigen, we will preferentially proliferate out or clone that one cell. The other guys in our repertoire don't get affected. They have to engage with their antigen in order to get cloned. Makes sense? So that's clonal selection. This leads to tolerating self-antigens. Sometimes if the signaling process goes awry in some way, we can still have, activation of anti-self clones. And this essentially activation of our forbidden clones, the ones that shouldn't have been activated that are anti-self. If we have the wrong signals, wrong time, can lead to proliferating those guys accidentally, and that's where we get autoimmune disease. B cells and T cells all come from the bone marrow, and then B cells do all of their maturation in the bone marrow. They stay in the bone marrow. And so then they will go to the lymph nodes, the spleen, and lymphatic tissues, and then they will do their filtering process there. Again, they're working at our checkpoints in our body. Once they've matured from the bone marrow, move to the checkpoints, and they wait there to engage with antigen, with their Ig receptors. Okay? All right, the T cells. T cells are first made in the bone marrow, and then they go to the thymus to continue their maturation. So the thymus is a gland in your chest. And so the T cells don't finish their maturation in the bone marrow. They only start out there as precursors, move to the thymus, and they're going to get their training in the thymus. A lot of that training happens very early on in your life, and so your thymus is constantly shrinking. But essentially, once they have moved into the thymus and they've been trained and matured from the thymus, they'll circulate throughout the body or hang out in the lymph nodes. They can kind of be in both places. They'll move around. So our cell-mediated immunities are all driven by T cells. So those MHC handles that we talked about earlier that say, I found something from outside or I found something from inside, right? Those handles saying, I found something from outside or inside. Those MHC molecules are there to interact with only T cells. Only T cells can recognize things being presented on MHC, because you have to have the captains activated before you can activate the B cells. B cells can see an antigen floating around, and on their little immune globulin receptors say, be checking at their checkpoints. I need to see your papers, please. They see your papers, they say, up, but I'm made to recognize black top marker. So sorry, hate to break it to you, but yeah, you're coming with me. But they can't do anything. They can take the antigen with them and then go ask the captain. They will show the captain and say, hey, I found black top marker. It's what I bind to. And so they have to find the captain that also recognizes only black top marker. And when they get the signal from that black top marker captain sends them cell signals. Remember cytokines? They're still cytokines. Special signals to activate that B cell that can then allow it to turn into a plasma cell and a memory cell and a regulator cell. All of our lymphocytes, once they get activated, will turn into the effector version, which is the one that actually does the job, the memory version, which is just going to wait around until it sees it next time, and the regulatory version, which kind of speaks for itself, right? It's going to make sure that our immune system doesn't go like haywire, right? So we always have three versions coming from all of our activated lymphocytes: memory, regulator, and then the big worker guy, the effector cell. So that can't happen. We can't make effector cells that make antibody. We can't have memory cells that hold on to memory later on. And we can't have the regulators unless we find the captain that matches our exact antigen and gives us the signal to go ahead and do that. Okay. So T cells, our cell-mediated immunity, is basically derived from our interaction with MHC. And our antigen presenting cells can present things on MHC2, stuff coming from outside, or any of the cells in your body can present on MHC1, asking for help, saying something's inside, right? So it's getting into starting all this stuff. Again, T cells will be either, essentially T cells can have memory cells, effector cells, and regulatory cells, just like we said. But there's two different types of effector cells in T cells. The helper cell, which is our captain, and the cytotoxic cell, which is like our tanks or our assassins. So those guys are going to be going out and actually killing infected cells. Those are the cytotoxic. It says it in their name, cell toxic. We're going out and killing cells that are infected. That's what cytotoxic T cells do. They do not kill bacteria. They do not kill viruses. They do not kill pathogens. They kill self cells. That's what they do. You have a viral infection, your viral infection will be controlled by your cytotoxic T cells responding, because you've got an internal inside of your cell infection going on. Your body is literally going out and killing all of those self cells that are infected. All right, we also have the regulatory ones that make sure, hey, if we're activating a cell that kills self cells, you better be sure you're killing the right things for the right circumstances and not just killing self cells non-discriminately. They better be holding up the right antigen, and they better be holding it on MHC1. And you also better be also getting signals from the captains that control all of this to say that you can do that, because otherwise you can see very easily how this is going to get out of control. There are some actions in your body that you're used to thinking of as, allergies or deviations from normal immune response that are driven by this cell-mediated sort of aspect. So anybody here know about poison ivy? Ever had poison ivy? Yeah, okay. Poison ivy is driven by your cytotoxic T cell response. So it's an oil that gets on your skin. There's an oil that interacts with your skin, and your cells think they're infected inside with something. And so they present the poison ivy oil on MHC1, and your cytotoxic T cells say, yep, I recognize that oil, and they go out and start killing your cells. You ever notice the difference between when you get hives from an allergy, that's just itchy hives, versus the blistery, nasty gooiness like what you get from, poison ivy? Big difference, right? Yeah. When you have that kind of an itchy, like actual damaged skin allergy going on, it's your cytotoxic T cell response. Yes.
So I'm highly allergic to poison ivy.
Yeah.
It puts me in the hospital.
Yeah.
But why? Why I can't just have like a little rash and be done with it?
Right. So
That's
your
my skin
cytotoxic T cell response is reacting to it with this going on, as well as probably also, you probably have a super antigen response. Like we were mentioned previously in the chapter, your body sees it as a super antigen. So it's overreacting on top of just the regular reaction. So everybody else that gets the itchy blistery rash, which is horrible enough, right? But yeah, everybody else that gets the itchy horrible rash that's associated with that, their body's responding to it normal CD8 response. Your CD8 response, your cytotoxic response, you get that, and you probably have a super antigen. You're getting too many signals.
Like every cell in my body.
Yeah. Yeah, it's activating your entire immune system instead of just this like focal sort of situation.
And it's not just poison ivy,
all of those, yeah, because they're usually going to go hand.
I was gonna say, I have a family member who's like the opposite, where there's no reaction at all.
Yep, I had my ex-husband he could go roll around in poison ivy and not get anything. But his genes, when they reassorted to recognize certain antigens, his just reassorted, so he didn't have any that react with that. That's all it is.
So it's not actually anything bad in the plant that's hurting you, it's you.
Yeah, it's you that's hurting you, yeah.
Okay.
Yeah. And so that's going to be a chapter 17 discussion again, when your immune system doesn't react the way that it's supposed to. We can talk about how great and wonderful and how much variety we get in our immune system, but that also ties into things like reacting to pet dander, reacting to poison ivy, things that aren't necessarily actually causing you a problem. Now, the plant has that as a defense mechanism. It has developed that because when it developed this oil, it stopped getting eaten by animals as much, because animals that ate it would have this allergic response and then know not to eat it. It didn't conscientiously make an oil that activates the immune system. It's not that smart. It just happened to make an oil that then animals stopped eating it. And that was a benefit to it. Yeah. Yeah, yeah. Yes, yeah. Yes. And so what can you do about that? And so, yeah, yours treats it as a super antigen. You get overstimulation, not just probably of your T cell response, but also probably of your antibody response as well. So a lot of times with our allergies, we can also have both things going on. Anybody in here have an allergy to metals? Any certain kind of metals? I mean, like sensitivity to like cheap metals or, yes, okay. So I buy some earrings, if they're not hypoallergenic, I will have an allergy to it. And so I have to have like gold plated, or I have to have like hypoallergenic metals that don't have nickel in it specifically. So nickel is extremely common to be allergic to. Usually nickel, it activates both sides of this. You get antibody against nickel and you get cytotoxic T cell response to nickel. So sometimes I'll just get hives that are itchy, and other times I'll get the weeping, like my earrings often usually get like real weepy and itchy and that's usually the CD8 cytotoxic T cell responses there. And so that's two different kinds of allergies that you can have. One of them led by your antibody response, and one of them led by your cytotoxic response. Okay? So that's the problem with being able to interact with a whole bunch of different antigens. All right, so if you have the CD4, this is how I remember this. I was about to write on the screen with this dry erase marker, you guys. Okay, if you have CD4, to me, the number 4 looks a lot like a certain letter to me, which is a capital H. There's a reason I'm bringing this up, okay? And then we have CD8. 8 is very round, and another letter that's also very round is C, okay? So this is how I remember this. 4 looks like an H. These are our helpers, also known as our captains. All right. C for cytotoxic. Cells that have the CD4 marker are going to be our helper cells. These are our captains that are going to be basically allowing the other responses to go. So what essentially will happen is you see an antigen and it is presented on some kind of MHC. This interaction with antigen and MHC allows our CD4 T helper cell to then help activate your B cells or your cytotoxic T cells. So this has to happen this way, that you have to activate the captain first. Because these guys can't do their job, the B and the cytotoxic T, they can't do their jobs as effector cells in your immune response until they get the go-ahead from the captain. So this is one way that we regulate our response. Okay? But we have to be interacting with MHC. So if we have antigen plus MHC1, where did the call come from? Inside the house, right? Or inside the cell. And then we will also have antigen plus MHC2. Where did it come from? Outside the cell. And it's presented by only APCs, right? All right. So these are our two possible responses, our inside cell and our outside the cell. Let me get rid of this captain first, so we have some space. All righty. So antigen plus MHC1, that MHC1 engages with a T helper cell. And because it is MHC1, the T helper cell can then be favored towards TH1, and TH1 interacts with cytotoxic T cells. This is your inside response. Okay, so 1 is this helper cell. From outside of the cell, we will activate our T helper cell, our captain. Because it came from outside, that captain will then be T helper 2. And this is going to activate your B cell, which is going to make what? Antibody. So this is our outside response. And look at that. All right, we're starting to follow how we're going to regulate our responses now. Our two different types of captains. TH1 favors the cytotoxic response. If you see something presented on MHC2, that T helper cell will favor the antibody response. Can you have both going on at once? Absolutely, and you will have both going on at once typically. If something is entirely outside stuff, it's really going to favor that Th2 response. But if you have a viral infection, viruses aren't only inside of the cell, right? They're also going to bud out and then be outside the cell sometimes. So you want both responses in a viral infection for sure. And anytime you have a bacterial infection that goes on inside of the cell, those happen as well. So, for example, tuberculosis lives inside of macrophage. Don't you love it? So tuberculosis lives inside of your macrophage, your antigen-presenting cell. But we need to make sure we're inciting that inside response to that sort of situation, but also inhibiting any of the outside bacteria from getting inside. So we need antibodies that prevent that from happening. So you usually have both going on, but based on how much more signal you get from inside or how much more signal you get from outside can basically favor one response or another more. And that's how your immune system is going to regulate to respond appropriately to those antigens. Are we following okay? Do we have any questions? Okay, so we've already talked about the T helper cells. The only ones you need to worry about in my class are Th1 and Th2. There are other ones involved in regulating and controlling all of those things going on in the background. I'm not concerned that you understand that stuff. I really want you to be sure that you understand the Th1 versus the Th2 and how they're governing the responses. Yes.
Sorry, I just think of the Th1 being the cytotoxic response.
Yes.
Okay, and then Th2 is the antibody.
Yes. And so if you ever have an issue with that, remember that the Th1 and the Th2 derive back from the MHC1 and the MHC2 respectively. So the reason it's called Th2 is because we got signals from MHC2. And if we can remember that MHC1, the one looks like an eye and that came from inside, then you can follow, okay, if it's inside, then you would want the cytotoxic response. But if it's outside, you would want the antibody response. All right. All right, so there's a video there. I'm not going to show it now, but if you want to learn more about how the T helpers regulate this stuff, there is a video for that. Okay, so when we start activating our cytotoxic T cells, again, 8. So we have CD4s, we have CD8s. Those are just markers on those cells that let us know which type of cell, T cell they are. The 8 is roundness like the C. The 4 has the harsh lines like the H's, okay? So our CD8 cell, basically, let's say your cells are infected inside with something, or you have an antigen cell that got infected inside of something. So if you have any sort of a cell situation where you got signals saying inside was infected, we are saying our MHC1 marker is holding out saying, help, help. I found antigen. I don't know what it is, but I have something I found that doesn't belong in here in me. So MHC1 screams out, we have antigen here from inside of a cell. Then the T cell receptor on the CD8 cell, only CD8 cells are going to be getting engaged with our MHC1 here. So this is where this gets a little bit complicated. This is where I hate having to delineate these things, but okay, so if it came from inside of the cell, MHC1, the CD8 cell will come and interact with that. That CD8 molecule that defines it as a CD8 cell recognizes MHC1. I'm going to draw it out because that we're starting to get the gist of the basics of it. All right, we've got an inside infection. This is important that we are remembering this is just for inside, okay? We have antigen, and it is bound on MHC1. All right. And it is inside of a cell, it's whatever, but it's bound on MHC1, and that this complex here being presented on the surface of the infected cell, that a T cell will come along that is CD8 positive. And then that molecule, CD8, is on the surface of that, and it recognizes specifically MHC1. All right. This is the first time that this CD8 T cell has ever seen MHC1 presenting this antigen. And it has a specific receptor. We'll just put receptor. We'll put TR for T cell receptor that recognizes this antigen. So we recognize our antigen. And our CD8 marker says, yep, it's presented on MHC1. It came from inside. Can this T cell, cytotoxic T cell, go ahead and kill that self cell now? No. Because why? Because it needs the signal from our captain to say it can do that. So how did the captain get activated though? Well, from outside of the cell, we picked up on an antigen presenting cell. We had MHC2, and it was holding on to whatever the antigen is, the same antigen. It's the same. Okay. But it came from outside. That was when our T helper cell came along. It had a T cell receptor that recognizes this antigen, and it had CD4 on its outside. It's a molecule on the outside of itself that recognizes MHC2. So T helper cells, remember how I said T helper, the H looks like the 4, and that lets us know it's CD4. As it turns out, yeah, MHC2 interacts with only CD4, and CD4 is found only on T helper cells. Okay. And this is going to happen from anything that's outside. So now that we've determined that our, please, I don't want to discard it, but I already drew all this stuff and I can't keep drawing on it now because I accidentally clicked this. I love technology. I accidentally clicked the side button on this pen. Okay, let's see if I have a better picture of it and I'll draw on that. pH, yes, we did this. Yes, we did this. Not really. We did talk about this on remove. Okay, well. Okay, so the whole process, I'm going to start from the top. You have an antigen in the body, and it could be picked up by an antigen presenting cell. It's going to, because it picked it up from the outside, put it on MHC2. Here's your antigen. Then our T cell receptor recognizes that antigen, and the CD4 recognizes MHC2. And then that will activate our T helper cell. All right, now our T helper cell, based on whatever signals it's getting from outside of itself, can become TH1 or TH2. And you'll usually make both when you've activated it, but you might use them for different things. So our TH1 cell, when we have inside stuff, we will have any cell that found antigen in it, can then present it on MHC1. I need more space here. And then we have antigen presented on that from inside. This does not want to. And that this is recognized by CD8 on the cytotoxic T cell, and it has a T cell receptor that recognizes the antigen. And then this is what's going to activate this reaction here. All right, the same thing can happen from still outside. We haven't gotten to the antibody stuff yet. You have a B cell, and it has its little Y-shaped immune globulin, and it will pick up antigen. and it will get signals from the Th2 that's already been activated in that previous step. So T helper cells are always going to be activated in that MHC2 response. And this is important because you can't have inside infections of cells without having something outside of the cell first also. Things don't just only stay inside of the cell. So you're always going to have both going on. But if something is primarily outside, you don't need to drive that cytotoxic T cell response. So the Th1 helper cells that are helping with inside stuff aren't going to be important if you have only outside stuff. If you have outside stuff, the Th2 guys are going to be doing most of the heavy lifting there, okay? So we have two types of captains, the inside captains and the outside captains. And based on what signals you're getting and what cells you're interacting with and what markers are involved on the surface of them determines if you're going to drive your cytotoxic response versus your antibody response. All right, so we're getting now a little bit more of the detail there. Right, I know, it's a lot, okay? Okay, if you activate your inside guys, your cytotoxic T cell guys, you're on the inside side of things, and you're activating your cytotoxic T cells, we have received MHC1 saying we've got inside, our CD8 molecule is verifying that. We're holding up antigen, our T cell receptor recognizes the antigen on the MHC1, the CD8 recognizes that it is MHC1, and we're getting signals from Th1 cells, our captain cells, saying inside response is go. How does our cytotoxic T cell kill the infected self cell? Through perforins and granzymes, okay? We've already learned about granules and how we have signal molecules involved in granules. These are granzymes. These are enzymes that are literally just going to break down anything in a cell. Perforins perforate. They are poking holes in the self cell, and then the granzyme will be able to break down everything in that cell. So it gets in because we poke the holes with the perforins. So perforins poke holes, granzymes break down everything in the target cell. This happens almost instantaneously. It's once that cytotoxic T cell releases that stuff, if you watch videos of it in real time, that cytotoxic T cell comes up. It might be a massive cell. One cell comes up to it. It gets its signal from the receptor, recognize the antigen on the MHC1. The CD8 molecule recognizes that it is MHC1. It gets its signal from the captain, Th1 captain, saying that it's okay to go, and then releases the perforins and granzymes. It takes me so long to say that, but all of that happens like, and that cell will go just immediately. The second that it contacts it, it happens almost instantaneously. And so those cells are contacted and immediately they're dead. And so we just go around just saying, you're holding up the MHC1 with the antigen, dead. Holding it up, dead. Holding it up, dead. Holding it up, dead. Constantly in your body as we're fighting viral infections. Just killing anything that has that specific antigen held up on MHC1. Your cytotoxic cells can do that. So now you understand why it's so important that we have regulation involved in that, because we're just going around decimating cells one after another. So how do B cells do their stuff? An activated B cell will differentiate into memory, regulator, and effector. So back to that again. So the effector version of a B cell is called a plasma cell, and it makes antibody. And essentially that antibody is going to serve as its receptor to interact with the antigen. It's going to get its signals from a Th2 cell, right? So it's presenting on MHC2 on the B cell. It's presenting the antigen here, and the T cell receptor is this guy in here. We recognize that it's on MHC2 with our CD4 molecule, and then we're able to send signals to the B cell to activate it. When we activated our B cell, we'll get our regulator, our memory, and our plasma cells. The plasma cells will crank out antibody, and those antibodies will go and interact with the pathogen. All right, we'll also have memory cells that'll be waiting in the future to be able to be interacted with. So we've already talked about the heavy and the light chains. We talked about the variable region where we're interacting with the antigen, and the constant region that dictates the function. All right, as it turns out, the FAB, the FAB region, the part that makes the antibody able to recognize the antigen, is the variable region. And the FC, go figure, hey, it's kind of already in the name. FC is also the constant. There's other names for these based on how they crystallize when we image them for pictures to determine the structure of how it works. All right. Let's talk about the functions of what our antibody does. I don't want to really get into, we talked a little bit about optimization. I want to talk about into all of our antibody functions. They can coat the surface, like it said in our other video with complement. Complement was able to coat our surface. It was like being covered in a bunch of flies. Same thing with our antibody. Our antibody can coat surfaces. It's like being covered in a bunch of flies. We can't interact with other bacteria. We can't engage with the surfaces of cells. We can't adhere to things. We're covered in an antibody. So coating is one thing we can do. Opsonization, providing a molecular handle for our phagocytes to grab onto. We couldn't grab onto the slippery surface of the bacteria. Now we have a handle to grab onto. That's opsonization. Neutralization. This is when an antibody interacts with a very specific receptor on the pathogen. So we know our spike proteins act as keys to get inside of our cells. So the spike proteins on viruses act as keys to get into the doors, certain doors on our cells, right? And so they mimic things to allow our cells to let them inside. Well, antibodies can bind very specifically to those spike proteins. That specific interaction of blocking the entry is neutralization. So there's coating, which is just like a physical prevention of function. Neutralization is specifically blocking docking, basically. Agglutination. Agglutination is what it sounds like. We're going to cause the pathogens to basically stick together by sticking all across a whole bunch of guys and causing them to kind of clot together. Then we have activating complement. You remember this. The classical pathway of activating complement is activated by antibody. So presence of antibody can activate complement. Start poking holes in these guys and they bleed out and die. And we have antitoxin. We don't bind to the pathogen, but the toxin that it makes. And that's what our antibody response does to protect us from tetanus, for example. So those are our 6 functions of antibody. Our classes of antibody, the first ones that we always make are IgM. And it's this pentamer up here. It doesn't have as many functions, but we can make it right away. The big workhorse antibody is IgG. Again, 80 to 85% of your total antibody in your serum will be IgG eventually. Once we switch over to it, it's going to be doing most of those 6 functions once we get them going. IgA is a secretory antibody. So not secret like you tell secrets, but secreted. So this is going to be associated with mucosa. So this is present in the mucus and not in the bloodstream. Then we have IgD, which is just the receptor that's found on the surface. And then IgE, which is mostly associated with allergies in the first world countries, but parasitic infections in third world countries. We talk about titers. Titers are concentrations of antibodies in your serum. And it can be measured over time to determine our response. We learned about this when we learned about ELISA. Whenever you first come into contact with a pathogen, it takes a long time to get your lymphocytes activated. It can take a week to get your lymphocytes actually fully activated because we're going through our innate response, our neutrophils going in and eating things up, then our macrophages going in and cleaning up, then those macrophages going and presenting it to our captains, our TH cells, then they have to get activated, and then they can help activate our B and cytotoxic T cells. And there's all this proliferation and control and regulation that's involved in each of these steps. It can take weeks in order for us to build up the immunity, and it can take weeks for it to start to plateau after that. Once you've seen it though, we have our memory cells in reserve, and they can get activated immediately upon seeing it again. And so that response can be next to immediate whenever we see that. So our primary response, we have a latent period because we are trying to activate our lymphocytes and get all of our regulation in gear. And at first, you're going to make IgM, and then we're going to switch to IgG. All right. In your secondary response, once you see the antigen, your response is immediate. And then look at what you're making most. This is IgG. You can make it right away. You don't have to wait on your IgM and then get IgG. Your memory cells hold on to that IgG. You can start cranking out IgG workhorse antibody immediately. And that way you're protected immediately and not over time. There is a video on here to overview our immune response. If you guys want to catch up on all this, because I know it's a lot and I don't have a lot of time to go and review all this stuff. We will have a review session on Saturday. I know we usually have them on Fridays at 10 a.m. I have a funeral that I have to go to on Friday. So I can't do it on Friday. So we're going to do it on Saturday at 10 a.m. And I will review the entire immune response on Saturday. So if you want to join in on that or watch the video recording of it, I will have an overview of the entire immune response from start to finish now that we've introduced this stuff, okay? We've already talked about active versus passive and natural versus artificial, so I'm not going to get into that. And then when you're designing vaccines, you can design vaccines against the whole pathogen or parts of the pathogen. And eventually you could use the live version of the pathogen or a killed version, and we can have all of these things we can add in called adjuvants that can help ensure that we are activating the response at the level and appropriately that we need. Because you don't want to put in a vaccine that only creates, that will have only a CD8 response and you get no antibody response. And that will have to allow you to get infected. You want to prevent the infection from happening, so you want to block the outside stuff. So a lot of times we have to help our immune system develop the response that we need, and adjuvants help with controlling our response in that regard. So adjuvants essentially work the same way as our cytokines do to drive how we're responding to antigens. Not all of our immune response, our memory, doesn't last forever. Those cells, they're not immortal. So at some point, those cells might just die off, and if they're not getting enough stimulation to keep that immunity going, you can just lose immunity. And that differs from cell to cell and antigen to antigen. So sometimes our immune response might need more boosters than others. And some things can be a lifetime of protection. Others need, boost every now and then. When we give our injections, we can give it subcutaneously, intramuscularly, and intradermally. Those are our different things. So subcutaneous means under the skin. Intramuscular means you are making sure that you're actually introducing it into the muscle tissue. Intradermal means that you're giving it at the surface of the skin. We used to give smallpox vaccines. Those were intradermal. We were like creating an excoriation of the skin and creating a scar. And that response was important in order to have the appropriate immune response and protection that was needed for smallpox. Those intradermal. We can give vaccines also through oral and nasal routes. We only do this with a few diseases, and oftentimes it has to do with allergies to what's in the actual injections. Those adjuvants, again, help basically drive the appropriate immune response, activate the antigen-presenting cells to make sure that we're getting all of the responses that we possibly can at the strength that we need them. When we talk about the COVID vaccine, someone was asking me why COVID doesn't protect as well as the MMR vaccine does. You get MMR vaccine, like you can literally walk up to somebody right now probably that has measles actively, and you would never have any symptoms or signs or fever, anything. And your body would be so quick to just power up its response against that, you'd never even know that got into your body, and it did get into your body. But with the COVID vaccine, in order to get our body to respond to it correctly, we had to make our body make the protein. How are we going to send signals during that whole process of waiting on your body to make the protein that helps elicit the right response? So we don't get the actual adjuvants that we needed to help elicit the proper response. So it's part of the reason why we don't have a very good response to the COVID antigen, because we couldn't add in the adjuvants that help drive the very strong response. The other reason is because the antigen itself is just not as strong of an antigen. So we just don't get as strong of a response. Remember when we talk about the strength and ability of antigens to drive the immune response there. So anyways, so we'll do chapter 17 and 18 on Monday. So we'll give abridged versions of those. So please be aware that you still need to know all the information that's in your study sheets for 17 and 18. But the core, I'm sure you can see now, 15 and 16, we really needed to focus on 15 and 16. 17 and 18 feed, well, 17 definitely feeds off of 15 and 16. 18 is a lot of just review of stuff we've already learned and just putting it into a diagnostic perspective. Okay, so let's go ahead and go up to the lab. And again, short day in lab today. We'll get that going.
Your 17 lectures when you were actually
Yeah. I get it. It's a lot. And you get 2 chapters on it. I took whole semesters, multiple semesters learning this stuff, so.
And so